Alright, I think we're going to get started. So, I kind of had a brain fart on Friday, and I forgot that I'm lecturing this week still. And so I kind of rushed a little bit through the large intestine when I kind of forgot that I could just incorporate it into this week. And I felt so bad that I didn't finish some of the material because it's like totally not my model. It's always finish my material. And so anyway, I realized this at 3 in the morning this morning when I was like, oh my gosh, I could totally just finish lecture that I didn't finish on Friday and then spread it out through the week because I know how long these next lectures are going to take. I've done them a lot of time. So anyway, so that's what we're going to do. I'm going to introduce the last topic because I feel like it's pertinent. It kind of like pulls all the information together that we've been going through. And then what we'll also do is I sent out 2 different links. One link is for you guys to tell me what times work to schedule review sessions. So I'll probably take the 2 most popular times and then schedule review sessions for those times so you guys can come. And then I've also asked and see if Lolly wanted to do a review session for the other time. So whatever the 3rd most popular time is, I'm going to see if she has availability during that time slot. So she can do a review session as well. That will at least give you a few different times to work with. I'll still have office hours on Monday, so if that time works for you, you can always still come to my office hours and talk to me about questions you have before the exam. And it is on Monday, so if you have any last-minute burning questions, that's fine. Come see me in office hours on Monday before, and we can get those straightened out. We can get to the last topic, which is kind of fun. I'm already losing my voice today. Okay, so can you die pooping? That's the question that I wanted to ask. And the answer is yes. Several people have actually died on the toilet. And this happens for lots of different reasons, okay? One is that when you actually have to go to the bathroom, there's this maneuver that's called the Valsalva maneuver that most people are probably familiar with if you didn't know it had a name though. This is basically you forcefully attempting to push the feces out of your body, okay? And typically you hold your breath when you do this. And as you do this, you get some different changes that you can see. You're pushing against a closed airway system, and so what happens is you get an increase in thoracic pressure. That bearing down process increases your thoracic pressure. And then this increases blood pressure, so what happens is you activate baroreceptors, which then in turn try and decrease blood pressure. Now, typically you can only do the Valsalva maneuver for a certain amount of time, so when you release that, now all of a sudden you have this drastic fall in blood pressure, you're no longer like bearing down, and so now you're waiting for your baroreceptors to kind of catch back up. But during that period of time, what can happen is you can have decreased perfusion of the heart. So now your heart is less perfused with blood. For some people, if you're susceptible to a heart attack at that point, you can initiate a heart attack. And this actually happens to people. It's more common than you would think. I looked up a list of like all these different famous people who died on the toilet, and there's a pretty big list. Elvis is probably the most famous. He died in the bathroom, and they think he had there's lots of reasons to believe that he was straining to go to the bathroom, okay? And that this could have induced his heart attack. He didn't have a heart attack. Some of his problems were that he was obese. Okay. So if you have been clogging in your arteries, and now your heart arteries are clogged, now you increase the perfusion of your heart, you're going to get less blood, less oxygen, you can induce a heart attack. Smoking, stress, he was a chronic drug user, he had high cholesterol. All of these things are contributors to, or factors for the onset of heart attacks, and he had all of these. And in addition to this, he had one other thing that really, I think, a lot of people don't know about him, but probably in some ways pushed him over the edge, and this is this Hirschsprunger's disease. And so we'll talk about what that is and why this could have even contributed more. So if we take a closer look, what happens in this disease? Well, here's a picture of what it kind of looks like. First of all, you will see that another name for this is megacolon, and you'll kind of see why that is. You have a huge enlargement of the colon. And the reason why is because you lose, these people are kind of born without nerves in the distal part of their colon, okay, and rectum. So now I want you to think about what we talked about with the short reflexes and the long reflex, the defecation reflex. What we have to be able to do is activate nerves that can relax the internal anal sphincter. Okay, if you cannot activate those nerves to relax that sphincter, you are going to have a much harder time going to the bathroom. And so if you don't have those nerves, your internal anal sphincter is tonically contracted and you can't relax it. Okay? And so what happens is you get backing up of feces that has a harder time moving into the space here. And also, think about all these other nerves that are helping with motility, helping with peristalsis, moving the feces forward. If you're missing those nerves, now you're unable to do that. Okay, so the feces kind of accumulates in the distal part of your colon, which is what happens. And so another thing is that Elvis was on a really high dose of laxatives. So when people have this disease, they typically go on a high dose of laxatives to help push the feces through or make it easier for it to pass. But a lot of times it's difficult because to go to the bathroom, they really have to strain and kind of push that sphincter open, which is not an easy task to do. And so when you combine all of those ideas together with Hirschsprunger's disease, you can kind of see how Elvis might have been even more susceptible to a drastic decrease in blood pressure, decrease in perfusion of the heart, and then in the end death. So I think that's all I wanted to say for that. Hopefully that kind of ties in the reflex piece with a disease state. And the other thing I wanted to say that I didn't say in class, and it's part of your learning objectives, is that the parasympathetic branch, so your sacral nerves that come back and help you do the relaxation piece, they're going to contribute more than, and the peristalsis. So those nerves help you with movement, and they're going to help you with the relaxation. Those nerves that come from the spinal cord are more important than the stretch reflex that's available to you. So if you lose those nerves, it really is going to contribute more. You're going to have way harder time defecating versus if you lost for whatever reason the stretch reflex and now the short reflex, and you still had the long reflex, to be way easier. So the long reflex contributes more than the short reflex, but both are happening at the same time. Okay. Somebody else also asked me what, sometimes there's a little bit of a difference between the learning objectives that I show on the screen and the learning objectives that are outlined. They're really all the same. Sometimes I just shorten them on the screen because otherwise I'd have a page of words that isn't always helpful. So I cover all the same material in the lecture. So if there's a learning objective that is on the piece of paper that you have in your course packet, I should cover it. That should all be covered, even if the exact wording isn't on my screen initially. So don't get confused by that. So, all right, let's move on. Also, the second survey is all of the learning objectives for the GI tract. And I just wanted to know from you guys kind of what you thought of what you picked two learning objectives that you had most difficulty understanding. And then if we have time, I'll try and budget in time throughout the week so we can cover a couple of those pieces. Just so that I know what you guys are confused about, and then I can address it. If you guys have both of those done by Wednesday, it's super easy. It's just a Google survey. It'll take you 5 minutes, not even. But it will really help you guys, and it'll really help me. Okay. All right, so now we're entering the metabolism piece. And we spent a lot of time talking about how do you get nutrients from the intestine into the blood, but we really haven't talked about why do you do that, where does that go, where do we store these nutrients. We know that a lot of these things go to the liver, and that the liver is going to pick up some of the nutrients, but the blood goes elsewhere, and those nutrients are picked up elsewhere, so that's kind of what we're going to talk about. And so it's kind of most fitting that we start with the absorptive state, because it kind of fits nicely with what we just talked about. You just ate a meal, you remove those nutrients into the blood. Now we're going to talk about what happens to those nutrients once they're in the blood. And then on Wednesday, we'll come back and we'll kind of look at the recall of those nutrients. So we store a lot. We store a lot as fat because it's really important. We're not eating constantly, so we needed a way to kind of call upon nutrients. And most of our nutrients are stored in the form of fat. We have a little bit of a sugar storage site, which we'll talk about. And so Wednesday we'll talk about calling on those nutrients when we don't have food around. And then on Friday we'll do thermal regulation. And yeah, that's where I will leave you guys, and I'll hand you off to Dr. Wu to close the course. Again, tomorrow there is 10 points of extra credit in the lab. So if you are in there, it's to your advantage to come and participate. Okay, so today, what happens between food, digestion, and storage? So why do we even eat food? I think that's a question that's worth asking. We eat food because we pull nutrients out of the food that we eat, and we use those nutrients to make ATP. Okay? That's the whole reason we eat. We need to have a currency that we use in the cell, and the currency in the cell that we use is ATP to help us do work. And so when we eat, whatever we eat, we can pull nutrients from that, for the most part, and we can convert those nutrients into ATP, which is the currency of our cell. Okay? And this makes evolutionary sense, too. We don't eat all the time. We don't have to constantly be eating, which is really nice. Normally there are periods of fast and famine, so like there were times when there's really abundant food and we could eat a lot of food at that time. And then we might go through a period where we go 2 or 3 days without eating. And that's fine too, because we have the ability to store. And we can call upon those storage sites. We know how much fat can be stored in a person. A person can increase their fat stores 3, 4, 5, 10 times. We can increase our body weights. I can double or triple my body weight and still survive. So we have this massive capacity to take on weight and massive capacity to store excess nutrients that we take in. And evolutionarily that makes sense, so that we don't constantly have to be looking for food to keep us alive. Okay. So here are our learning objectives. We're basically going to be talking about the movement of these molecules from the blood into their storage site. We'll talk about glucose initially, amino acids, how they go from your intestine, from the gut, to the liver, and then the peripheral cells where they're used and stored. So these are kind of themes that are going to come back. We're either going to use it or we're going to store it. We don't waste it. So when you finally get nutrients in your body, you're going to either use it or store it. Okay? And we'll talk about these different processes, glycogenesis and lipogenesis in the liver, and how they prevent large spikes in plasma glucose after a meal. This is another kind of important theme that we'll talk about. Glucose in your bloodstream is bad. It's toxic. So we don't want to keep elevated levels of glucose in our bloodstream. That's why people with diabetes have such problems and other complications as a result of having elevated levels of sugar in their bloodstream. So we want to do all that we can to prevent huge spikes in glucose. Okay? And these different pathways kind of help us do that. Okay? And then we're going to contrast storage of absorbed triglycerides. What I mean by that is triglycerides that come from your diet into your bloodstream, and compare those to triglycerides that are synthesized by the liver. And we can take glucose and make triglycerides. We can take existing fats that come from our diet, repackage them into triglycerides. We can take amino acids if you have them in excess and make them into triglycerides. Those are things that are done by the liver, and we'll kind of compare and contrast that. Then we'll talk about the most important hormone in the absorptive state, which is insulin, and what it does, and how it really plays a central role in all of the reactions of the absorptive state. And then lastly, we'll talk about diabetes and hyperglycemia, and that's kind of where we'll end. All right, so metabolism is the sum of all the chemical reactions in your body. All right. We extract energy from nutrients, okay, and then we want that energy to do work. And we make ATP, and we use that to do things like contract our muscles, to make sure our heart can beat all the time. And if we have excess, then we store it. There are two major pathways that dominate during the post-absorptive and absorptive state. We have anabolic pathways, and I always think of these like anabolic steroids, like you're building something, you're building muscle, you're building up a compound from smaller subunits, okay? So anabolic pathways synthesize larger molecules from smaller ones. An example of this would be glucose molecules. We talked about glycogen, how it's like kind of like a chain link fence, right? You can take individual glucose molecules that you've eaten, and our liver can hook them together and make a longer glucose strand called glycogen. And that would be an example of an anabolic pathway. Okay, these pathways tend to dominate in the fed state. Okay, we store glucose as glycogen, so we want to create glycogen and we want to store that. Okay, we can synthesize fatty acids from glucose, so we might make these long fatty acid chains, and we'll store those as triglycerides. Okay, those pathways tend to dominate in the fed state. Catabolic pathways are kind of the opposite. They break down large molecules into smaller ones. Okay, these tend to be dominant in the fasted state. We might take glycogen, break it down to individual glucose molecules so we can use it for energy. We'll break down triglycerides to fatty acids so we can use it to make energy when we don't have food around. It's usually when we call upon our storage sites when those pathways are active. So I've kind of listed here for you the two, what I think of the two major pathways, and what the pathways that you guys need to know about and where they fall. So for anabolic pathways, pathways that build things up, okay? We have glycogenesis. Okay? Glycogen, or glyco for glycogen. And genesis means the formation of. So every time you see genesis, I want you to think of, I'm forming something. So you should automatically assume that it's an anabolic pathway. Right? And glyco means glycogen, so these words are telling you something about what they're actually doing. So try not to memorize them, try and interpret them. What do they mean? They're telling you exactly what they mean. Lipogenesis. Lipo for lipids. Genesis for the creation of, so you're forming new lipids. Then we have gluconeogenesis. This one sometimes is harder. It's okay. Gluco for glucose. Neo means new, and genesis means formation of. So what the body is doing is you're forming new glucose. And we'll talk about this on Wednesday more. But the idea is that you're taking a molecule that was once nothing to do with glucose. Something like pyruvate. Something like lactic acid, something like an amino acid. And you're converting it to sugar. Okay, that's why we're synthesizing new sugar. This isn't glycogen, that's already a bunch of sugars put together, and we're cutting off individual glucose molecules. The liver is doing a very special job converting these substrates that didn't really have anything to do with glucose before and converting it to sugar. And you'll see why that's really important in the post-absorptive state. Okay, then we have catabolic pathways, which is glycogenolysis. Now, lysis means to break apart, to break. Okay, so when we have glycogen, that's for glycogen lysis. You're breaking glycogen up. And what you're doing is you're creating individual glucose monomers, you're putting those in the bloodstream. That's a breakdown of glycogen. Lipolysis, lipo for lipids, breakdown of lipids. All right, so these should intuitively make sense to you both, or to all of you. All right. I've highlighted here which ones are dominant in the absorptive state and which ones are dominant in the post-absorptive state, so you can have generally a good idea of what's going on. Notice that this is a formation process. This is happening in the post-absorptive state. When sugar levels are low, we need a way to make sugar, to put it in the bloodstream, ultimately to feed the brain. Okay, and we'll talk about that on Wednesday, so don't get hung up. All right. Again, I'm going to bring up this theme of you're going to use it or you're going to store it. So ingested biomolecules have three fates. You're going to use them to make energy to do work. We have things that need to be done in our cells, phosphorylation events that need to happen. With like muscle contraction is a perfect example that consumes massive amounts. The sodium potassium ATPase, one of the largest consumers of ATP in our bodies. We take in nutrients every day so that we can pump that pump. I think a third of our energy goes to just pumping those pumps alone. Okay, so a huge amount goes to that. When you're eating a lot, it makes sense that you would have growth and maintenance going on. Okay, you can develop new cells because you have lots of ATP, you have lots of energy that's available to use. When you're fasting and you're starving for a week, does it really make sense to be synthesizing new cells? Probably not. You're going to go into more of a mode where you're conserving as much as possible. Your metabolism is going to fall. All of those things. So when you have food available, you're going to use it. You're going to help synthesize new cells. You're going to grow. And a lot of this goes to maintenance. And you'll find out about this on Wednesday too, but when you are in a starving state, a lot of what you're losing is fat, but most of what you're losing to make sugar is muscle. Okay? And so when you eat again, the protein that you eat goes to replenish the muscle that was lost previously. And then if you have excess, you're going to store it as glycogen or fat. Now, the nutrient pools that are available for you to use. These are the macronutrients. Fatty acids, glucose, amino acids. This is what you eat in your diet. And these are the things that your body's trying to extract from that. Fatty acids, glucose, amino acids. That's what you're using to make energy. That's what you're using to survive. Okay? So, excess energy. These are our only two storage sites in our body. Actually, I shouldn't say that. These are the major storage sites and the ones that I want you to know about. Okay? Glycogen. This is our major way of storing sugar. This sugar reservoir is tiny. Okay? We don't have a huge glycogen storage site. And the reason is because glycogen is extremely heavy. Glycogen needs to be hydrated, so it needs to stay dissolved in the cells. And then because it needs to be hydrated, it carries with it a lot of weight. Okay? In fact, it's about 10 times heavier than the same amount of energy that you can get from fat. So it takes about 2 pounds or 1,000 grams of potatoes, which is similar to glycogen. Okay, so if I had 2 pounds of potatoes here, and then I had 90 grams or 10 times less olive oil, which is similar to what we store for fat, they have the same amount of energy potential. So if you burn that amount of oil, you get this amount of energy. You burn that same amount of glycogen, you get the same amount of energy. So if we carried everything around on our bodies as glycogen, we would weigh so much more. It'd be hard for us to move around. We wouldn't be able to chase after things and kill animals that we would need to eat. It would just be, it'd be very cumbersome. So we developed this way of carrying around a much lighter, more efficient energy source, and that's why we have fat, and we use fat as our energy source. Okay, so they have almost, I originally had twice as much, but I was just reading about it, and it's actually more like 10 times. Probably depending on the type of fat that you're looking at. But the content of energy is equal amounts of carbohydrate. Okay? The energy in fat, though, is slower and harder to access. It's stored in the adipocyte, in the fat cell, so we have to call upon it, and then it has to get moved out into the bloodstream where we then can use it. Glycogen is typically found in 2 spots within our body. It's found in the liver, and it's found in muscle. And in the muscle, it's because you need to run away from something quickly. You need a rapid source of energy, and that's your glycogen. Okay, that's about 20 minutes of intense running, and it's gone. All right? In the liver, it has a different job. Its job is to give you an immediate source of glucose for the brain. So the liver chops up the glucose molecules, puts it in the bloodstream so that you have access. So the brain has access to food if, for whatever reason, your sugar levels fall. Okay. So here's a diagram from your book. Kind of a busy diagram, I totally did it. We'll break it down individually, so don't worry. But I show you this because I think it tells you everything that you need to know about the absorptive state, or all of the main points that I think are worth understanding for the absorptive state, all in one diagram. And so we'll kind of break this down slowly. But the main ideas are that you have oxidation happening. You're producing lots of ATP from sugar and from other molecules too, you'll see. When you've eaten a lot of sugar, the body, when it's in the absorptive state, tends to burn sugar primarily. And this does a couple things for you too. And then if we have excess, you'll see that a lot of it gets stored here as a triglyceride, and so we'll kind of move through this. All right, so let's break this down individually. Okay, here's Gus again. He's eating the spaghetti. we'll start with carbohydrates. So we'll start with the spaghetti strands. And that's the other thing that I said glycogen stores are really small. So when you carbo load, that's the whole idea behind carbo loading, right? If you want to fill your glycogen stores. Your glycogen stores are really small. So do you need 5 plates of pasta to fill your glycogen stores? No. You actually probably don't need that much. And most of the time, your glycogen stores aren't completely depleted or empty. They're already kind of halfway full. So the idea behind carbo loading is to ensure that you have enough glucose if you need to tap into it when you need it, so you can have a rapid source for your muscle. But the idea of eating as much carbs as possible doesn't always pan out. But we're going to start with carbs, so let's kind of see what happens. Okay. So let's talk about the different fates of glucose. Here's glucose highlighted in this image. I can't get rid of these numbers, so just ignore them, all right? We'll start with number 1, but this won't necessarily go in order. Okay, so most of your tissues, when glucose levels are high in the blood, remember it's toxic. We don't want that. So we need ways of getting it out of the blood. How do we get out of the blood? Okay? One way is have all of your tissues start pulling it out of the blood and oxidizing for ATP. That starts lowering glucose levels. That's a good thing. Okay? So all of your tissues start oxidizing glucose, including the brain. Everyone's breaking down glucose to make ATP. That's what I mean when I say oxidizing. Number 2, glucose can go to the skeletal muscle and can be stored as glycogen. That's that small reservoir. Right? Muscles also burning it for ATP as well. And then number 3, glucose can go to the liver, and when it's in the liver can do a couple different things with it. Number 1, if the liver also has a glycogen storage site, so it will make glycogen. The liver will also take it, and it can make triglyceride. Okay? So this glycerol 3-phosphate is basically a 3-carbon backbone. I will show you a picture. I wish I should have had a picture up here. But basically, you need this 3-carbon backbone to attach your fatty acids to them. So you have triglyceride as your backbone with 3 fatty acid tails. Okay? So this is basically glucose being converted into that carbon backbone. And glucose can also be made into those fatty acid tails. So basically what you're making is a triglyceride. So when you attach that backbone to 3 tails, you have a triglyceride. So your liver is busy making lots of triglycerides from the excess from the glucose that you're pulling in. Now, this process of making triglycerides helps lower glucose levels in the bloodstream. You see how all of these things are pulling glucose out of the bloodstream so it's not elevated for an extended period of time. That's healthy for you. That's good for you. And triglycerides are good because they eventually will get stored in the adipocyte, in your adipose tissue. And then when you need it, when you're not eating anymore, you'll call upon those nutrients. Okay? So the 3 fates of glucose: oxidation, burning it, using it to make ATP. That's a good thing to do. You're going to store it as glycogen in the liver and muscle. These are small amounts of glucose that are being stored here. Triglyceride synthesis, the vast majority is going to be made into fat. So maybe you didn't know that if you just eat spoonfuls of sugar for dinner and you think, oh, it's fine, I'm not eating fat, well, all of that excess sugar is going to be converted into fat anyway. And so it's pretty much like eating a spoonful of fat in some ways. But your body has to convert it to fat first. Okay? Triglyceride synthesis can also happen in the adipose tissue. So the adipocyte has also the ability to take sugar and make triglycerides as well, and you'll see that in your diagram when I take that little square away. So know that triglyceride synthesis happens in the liver, also happens in the fat tissue as well. Okay, so let's talk about the meat. Proteins, amino acids, what happens with those? All right. Here's your amino acids. Here are the fates of the amino acids. Again, oxidation can happen. Now, primarily, all of your tissues are using glucose. Okay? The liver really is the only place that's using amino acids to make energy. Okay? So you can write that down. And that's what they're showing you here. Amino acids have kind of 2 fates here. They're being made into ATP in the liver. Okay? So the liver is taking amino acids and it's making ATP, which is a little bit different. Okay? The liver can take glucose to make ATP as well, but it's taking amino acids and doing the same thing. Okay, so that's oxidation. When you make ATP from something, that's oxidation. So the liver oxidizes amino acids for ATP. The amino acids also go back to the muscle. Remember, I told you when you're not eating, you lose a lot of muscle to help you make glucose. And so you need to replenish that muscle. So every time after you haven't eaten for a while, now you've fasted, now you've eaten again, now you've eaten some protein, that goes to replenishing your muscles. If you were to starve yourself to death, you would typically not die because you lost so much fat. You would typically die because you lost too much muscle. Okay? We sacrifice our bodies, and the whole idea is that muscle is used to make glucose when you're not eating. Okay? To feed the brain, care about the brain. So we will sacrifice our muscle for the brain every day of the week. The reason why you die is because you lose 30% of your cardiac muscle or 30% of your diaphragm, you're dead. Okay? But our bodies have this ability, we will sacrifice for the brain. We care so much about the brain, making sure that it has access to sugar, because it can't actually oxidize fat. So you'll see that in the next slide too. So you're breaking down lots of protein to help feed the brain. When you're not eating, that protein needs to be replenished. The amino acids that you consume in your diet go to also replenishing that lost protein. Okay? And then anything in excess is going to be made into triglyceride. Okay? So this is done also by the liver. Okay? So there's one pathway here, amino acids go to making ATP through the formation of keto acids. We'll talk about that later, so don't get too hung up on this. Okay? But in the liver, the amino acids also start making fatty acids and make triglycerides. So if you only eat steak all day, you can still store a lot of fat, but our bodies are going to be changing those amino acids into triglycerides for long-term storage. Okay. Yes, and then this step is called the deamination step. This will become more important in the post-absorptive state, so this will come back again. But I just wanted to point out that this is the same thing that's happening in the post-absorptive state. This changing of amino acids to a keto acid is called deamination, and we'll talk about a little bit more about that on Wednesday. But I just want to highlight it's the same thing. All right, so let's talk about fat. Okay, so these triglycerides here that are being synthesized have to go somewhere. They don't stay in the liver. If they stay in the liver, you get fatty liver disease, and that's not good for you either. That's a disease state. The liver isn't made to store fat. Okay, it's not made to handle lots of grease. But our fat cells are made to handle lots of grease. They have a very specialized storage site for those fats. And so we really want to push all of those triglycerides to the adipose tissue for long-term storage. So let's look at triglycerides here. Okay, now this is where we start comparing and contrasting. We have triglycerides in the form of chylomicrons. Those triglycerides, remember from the digestive system, those come from what we eat. Remember, our intestinal tissue forms these little chylomicrons, pushes those chylomicrons into the lymph, and now the lymph eventually meets up with the bloodstream. Now you have chylomicrons in the blood. Chylomicrons only contain fatty acids, triglycerides from your diet. Okay, those came from your intestine, were repackaged in this specialized package called a chylomicron. It was pushed into the bloodstream. So the only fats that are inside of there are fats that you ate. Okay? So that's one type. The other place where you have triglycerides is here. These are triglycerides that are made by the liver. Okay? Notice the liver is making triglycerides from sugar. It's making triglycerides from amino acids. Okay? And so those are newly synthesized fats. Okay? And so the liver will package those fats up into a separate package, very similar to a chylomicron structure, but it's different. And it sends those out into the bloodstream. Now, chylomicrons and what you will see is VLDLs. That's the specialized package. They're very similar in characteristics. I'll show you kind of what they look like. They do very similar jobs. They basically unload their contents onto tissues. Okay? Unload their contents into the fat cells. So they carry they're huge blobs of fat. They dock onto the fat cell and they dump their contents into the fat cell, and the fat cell says, okay, I can store these for you long term. Okay. So the fates of a triglyceride are you're going to store it in the adipose tissue, or you're going to burn it. Okay? Burning a fat typically only happens in the post-absorptive state when you're not eating. And the reason is because you have sugar around, and sugar is easier to burn than fat. Okay, so we preferentially, if sugar is around, one, we want to get out of the bloodstream, and two, it's easier to burn, so we burn it in preference to fat. If we have fat around, we typically store it for times when we don't need it, or when we need to call upon it, when glucose isn't readily available. All right, I like to do this. I like to show the same idea in a different figure because sometimes some people interpret figures differently. This figure might speak to you more than the last one. I like this figure a little bit better. I think it breaks things down a little bit more, so I want to show you this one too. Anyway, so here you are in the intestine. Okay, so these are all of your dietary fats that you've taken in. You just ate a really fatty meal. Okay, and these bile salts are helping you emulsify that fat. You're releasing these fatty acids, phospholipids, monoglycerides. They get absorbed in the intestinal cell. This is a nice review for you guys, by the way, too. The intestinal cell then resynthesizes that fat and puts it into a ball of fat called a chylomicron. And that chylomicron moves into the lymph. Remember, it can't move into the blood. It's too big to move into the capillaries, so it moves into the lymph. Once it's in the lymph, it then eventually meets up with the blood, which is what I'm showing you right here. And now you have chylomicrons in the blood. So what does a chylomicron look like? This is kind of what a chylomicron looks like. And why the heck do we even do this? Why do we spend time making this huge ball of fat? You know, why don't we just have fats floating around in the bloodstream? Well, fats aren't soluble in water, okay? And so it's like mixing oil and water. So you wouldn't want that floating in your bloodstream. So what we do is we coat it with phospholipids, which are water-loving, okay? One layer. Phospholipids have these fatty acid tails, right? Fat likes to associate with fat, and so the tails associate with the fatty acids that you've eaten. Then they're happy to associate with each other. The polar heads associate with the water-loving side. Okay, so now you have this ability to kind of dissolve the fat in the blood. You increase its ability to dissolve the fat in the blood and mix it. So now you can carry these fats, and you're not going to mess with as much with viscosity and changing all these different properties of the blood. The blood can still flow pretty smoothly. All right. So you store these because you want to increase its ability to kind of dissolve within the blood, so that your fat isn't sticking to the sides of your artery and causing you atherosclerosis. Okay. All right, on the outside of these balls of fat, these chylomicrons, you have proteins, these apoproteins you'll see. Those are helpful because those are what signal to the fat, hey, I'm a ball of fat. When those apoproteins bind to their receptors found on adipose tissue, they stop and then they unload their contents. Okay? So anything that has an aporeceptor will allow these things to dock and release their contents into the tissue. Okay? So fat has tons of aporeceptors. Now, if you don't have an aporeceptor, those fats will circulate forever in your bloodstream, or not forever, for a really long time. You're susceptible to atherosclerosis. That's a very common mouse model that's used in the lab. They take away the apoproteins. Now these balls of fat just circulate in the bloodstream because they have nowhere to dock, and now these mice develop massive atherosclerotic plaques. It's kind of a cool idea. So, anyway, back to this. The chylomicrons, when you synthesize them, you put apoproteins in them. That's what that apo is. Okay, so these chylomicrons have the apoproteins. Those get sent out into the blood. Where do they go? Well, the majority of them dock onto adipocytes. Okay, when they dock onto the adipocytes, this lipoprotein lipase, it's a lipase. It breaks up the fat. Okay, so again, triglycerides are too big to move out and diffuse into the cell. So you have to break up that triglyceride into fatty acids. Remember, you have the glycerol backbone, that 3-carbon backbone, and you have 3 fatty acids attached. 1, 2, 3. You have to break those 3 fatty acids off, and now you have their carbon backbone. Both of those can diffuse in. Okay, so you have to break it up, and then you have to resynthesize it again. Okay, then you have to remake the triglyceride and then put it into long-term storage. That's why it's a cumbersome process, but it's the way that we've evolved to store lipids, okay, for long-term storage. Now, as you can imagine, as that little chylomicron is circulating, it's going to get smaller and smaller and smaller as it dumps its contents off, right? And so we call these chylomicron remnants. And as those remnants become smaller and smaller and smaller, they still have a little bit of fat inside of them. They haven't dumped everything. And so we don't want to waste that. What we do is the liver says, okay, I'll take those chylomicron remnants, I'll kind of consolidate them, take fats from all these different ones and put it into a new package, and I'll send that on to the blood and have it do the same thing over again. Okay? So it's like a recycling center, basically. basically. You have these chylomicron remnants that enter the liver, and the liver says, okay, I can take those chylomicron remnants, and what I'm going to do is I'm going to produce what we call a lipoprotein complex. Okay? Lipoprotein complexes, look, you wouldn't be able to tell the difference between a lipoprotein complex and a chylomicron if you looked at it under the microscope. They have different sets of proteins on their surface. They're very slightly, but their function is entirely the same. Their function is to dump fats into cells or into the adipocytes. Okay? So their function is exactly the same in terms of this class, but where they come from is where the fats originate is totally different. So a VLDL will contain fats that came from your diet through this chylomicron remnant pathway. They will also contain newly synthesized fats. So fats that were once glucose in your diet, but now are fats. Fats that were once amino acids coming from your diet, but now are fats. Okay? So knowing the difference between a VLDL, what does a VLDL hold, and a chylomicron, where do those fats come from, is something that I probably will ask you about. Okay? That's important. All right, but their function is exactly the same. These things dock onto triglycerides and they dump their fats into the triglycerides. Some cells oxidize fats for energy. That's very minimal compared to in the post-absorptive state. You'll see there's a switch. Okay? All of a sudden we start oxidizing fats like crazy. But when there's glucose around in the absorptive state, we tend to oxidize glucose and we tend to store the excess fat. Okay? So this is going to be happening way more often than not. Okay? Chylomicrons going into triglycerides. Lipoprotein complexes or very low density lipoproteins going into triglycerides for long-term storage is important. We are not going to talk about HDLs or LDLs. If you're interested in, we can chat about what they do and what the difference is. Why are they good cholesterol? What's bad cholesterol? On and on. We can talk about that, but that's not something we're going to cover in this class. So what I want you to know is what is a VLDL. I want you to be able to compare it and contrast it to a chylomicron. So, if you want to do that, where do the lipids originate is a good question to ask yourself. I want you to know what an apoprotein does. That's important. And where do the lipids end up? Okay, that's kind of what I think you should understand. All right, so we have insulin. This is a major hormone that you should know about in the absorptive state. It tends to activate all of these different pathways involved with storage and usage of glucose, okay? Storage of fat, using glucose. And so all of these pathways are highlighted here. I'm not going to kind of go through each individual one, but look how it has a role for carbohydrates, proteins, and fats. So you can kind of read through these. The idea is that insulin is a driver of all of these different reactions in the absorptive state. So where does it come from? Well, we've talked a lot about the pancreas. We've mostly focused on exocrine functions of the pancreas, producing digestive enzymes, putting it into the intestine, producing bicarb, putting it into the intestine. Now we're going to talk about its endocrine function. Endocrine means that it's producing a substance that's putting into the blood. Okay, so we have these specialized areas, these islets, that are groupings of cells. And here they're showing you that basically there's 3 different cell types in these islets. This only makes up 2% of the pancreas. It's a really small amount of the pancreas. But we know if you kill your beta cells, you know how to produce your disease, diabetes. Okay, so these cells are super important. They're the ones kind of shown here. And they secrete insulin. And insulin is, again, a major driver in the absorptive state. So what does insulin do? And what factors control it? So when is insulin released? Well, how does the body know when to release insulin? The most obvious answer is when we have lots of sugar around. We know it regulates sugar. So if you eat a sugary meal, your glucose levels rise. That's a perfect stimulus for insulin release, because that's exactly when you want insulin to be released. Insulin helps you lower blood sugar levels. So the primary stimulus, the most important, is an increase in plasma glucose levels. Second, amino acids. Those can also stimulate insulin release. Okay? Feed-forward mechanism. Okay? Anytime you see the word feed-forward, I want you to think of an anticipatory mechanism. So this goes again back to the GI. Okay, in the GI, if you have a piece of cake sitting there, there are these little sensors that sense, oh, there's a lot of sugar in the food I just ate. And they release a hormone called insulin. And they go back to the pancreas and they say, hey, I know that blood sugar levels in the blood haven't changed, or blood sugar levels are the same. But in the intestine, there's tons of sugar. It's coming to the bloodstream, so you're probably going to want to, in anticipation, release some insulin. And so that's a feed-forward mechanism. And these are called incretins. Okay, these are these feed-forward hormones that preemptively tell the pancreas to release insulin, so that when the glucose does hit the bloodstream, you already have insulin around. You can start getting it out. Okay. Parasympathetic activity, as you can imagine, also activates insulin release. This is in the fed state. That should make sense, and this should also make sense. Parasympathetic or sympathetic activity should inhibit it. Okay. Okay. So, what does insulin do? Insulin, this is a super simplified version. Okay. I spent 5 years studying this pathway, and so there's a lot going on. But the idea is that insulin, when it's high because glucose levels are high in the bloodstream, right? So glucose levels stimulate the release of insulin. Insulin will bind to any cell that has a receptor present, the insulin receptor. Okay, that means that tissue is sensitive to insulin. Okay? So what tissues are sensitive? Muscle, fat, liver. Okay? If there's this receptor around, insulin will bind, and it activates a pathway inside of the cell. Okay? And that pathway then leads to the fusion of these transporters, glucose transporters, to the plasma membrane. So, see how these transporters are sitting inside of the cell, waiting for that signal? When they get the signal, they move to the plasma membrane, and now you get movement of glucose inside the cell. It's really cool. That makes sense. And so what does that do? Glucose moving into the cell allows these cells to not only take in glucose and oxidize it for energy, but it lowers plasma glucose levels. It lowers glucose levels down to a normal range, okay, which is exactly what you want. All right, so insulin promotes anabolism. We kind of already talked about this. Increases glucose transport, enhances cellular utilization and storage of glucose. Just talked about that. Enhances utilization of amino acids, and it promotes fat synthesis. These are the primary things that insulin is doing, and these are kind of highlighted here. I won't take too much time. This is kind of the same, this is a flow chart basically that summarizes everything I just talked about. The very first picture that I showed you where I said this is kind of like everything you need to know, this is also a lot of what you need to know in words. So yeah, it's hard to read. But the idea is the same. So all of these different pathways are converging on insulin increasing so you can decrease plasma glucose. So initially glucose levels are going to go up. We're going to do a bunch of things to bring glucose levels back down. And there's negative feedback regulation. So I won't go through this. This is in your notes. You can kind of move through this. And it's everything we just talked about and just covered. All right, so in the last few minutes, we'll talk about diabetes. So there are two. So at first, I usually like to break down this word. So diabetes mellitus. It has to do with having sugar and a sieve, okay? And so this idea, people noticed thousands of years ago, like 3,000 years ago, that people who had this disease had sugar in their urine, okay? And so how did they know that? Why did they think it was sugar? Well, they noticed that people who had this mysterious disease, when they peed, their pee attracted ants. So back in like the ancient civilizations, like Egyptian time, 3,000 to 5,000 years ago, it was the first documentation of diabetes, where they noticed that people who peed tended to attract ants, and the ants were attracted to their sugar that was present in their urine. Now, they had no idea how that sugar got there. They had no idea that this sugar was originating from the food that they were consuming, and their bodies are also producing sugar in response to the disease. And they thought, they had no idea what was going on. But they noticed that there's severe wasting of their bodies. So this is a disease that was uncontrolled and you would die at a really young age. Okay? So diabetes means to pass through like a sieve. And mellitus has to do with the sugar. Okay? So there are two, well, and then then we fast forward like 100 years ago, or 1,000 years ago. So within like 4,000 years, this is how advanced they became. Instead of watching ants be attracted to it, they had people who would taste the urine. And if the urine was sweet, they thought, okay, you have this disease, and they were called tasters. And so people would actually, that was their job, was to taste other people's urine to diagnose them with diabetes. But then they didn't know how to cure it, okay? And it wasn't until the 70s when they actually discovered insulin in dogs, these experiments were done in dogs, and they won the Nobel Prize in physiology for this discovery that insulin was really what was missing in these people and these animals, and that was causing the glucose levels to rise. So really the correlation between insulin and glucose wasn't made until around the 70s. And so it's a lot of time had passed and a lot of people died from this disease because it was uncharacterized. Okay, so this comes in two types. Type 1 is characterized by insulin deficiency. That means that these people do not produce insulin. Okay? They don't produce insulin. So what happens if you don't have insulin around? Glucose levels rise, right? So that's type 1 diabetes. That's because they attack their pancreas and it dies. Type 2 is the insulin-resistant form. This is the most common form. This means that you stop responding to insulin. Even though it is around, you don't respond to it anymore. Okay? Both, so it's just like not having it there in the first place. Okay? Both are characterized by elevated levels of glucose in the bloodstream. Both are bad if uncontrolled. Complications, it affects blood vessels, eyes, nervous system, kidneys, all of these things. And we don't really have time to talk about it, but basically the glucose modifies lots of proteins inside of your bloodstream that can lead to nerve complications and lots of vessel complications. And if you have too much glucose in your bloodstream, what you'll learn about in the kidney is that you can't reabsorb all of it and you pee it out. So that's where the glucose comes from. Okay, that's all I have. I'll see you guys on Wednesday, and I'll see you guys tomorrow when we do the exercise phys class.